Welcome back to BiochemSerye Learning on the Go. For episode 7, we're diving back into amino acids.
Back to the lab, ready to unlock the secrets of all 20 amino acids, all the ones that make up human proteins.
You know, back when I was a student, memorizing all these, it felt like such a chore. But now, seeing how each one plays a unique role, is pretty amazing.
Absolutely. And it's not just for exams. Understanding these building blocks, it's crucial for grasping, all of human biology.
Okay, so let's unpack this. First, what makes some amino acids essential? They're like they're on the VIP list of nutrients.
Think of it like a recipe. You absolutely cannot substitute certain ingredients. Our bodies, they can't make these 9, so we got to get them from food. Threonine, tyrosine, valine, phenylalanine, isoleucine, leucine, lysine, methionine, and histidine.
No shortcuts. All right, now, onto a pH check. Acidic versus basic amino acids. It all comes down to their side chains, right?
Exactly. At physiological pH, acidic amino acids like aspartic acid and glutamic acid, they have negatively charged side chains.
Okay, those are easy to remember. Acid is right in the name. But the basic ones, how do you keep those straight?
Well, there's that mnemonic, her leggings are basic, for histidine, lysine, and arginine. But what's really cool is how this charge difference, it influences how they interact with other molecules. Imagine them like attracting or repelling each other, shaping that intricate dance of protein folding.
All right, now this is where it gets really interesting. Ketogenic, glucogenic, and mixed amino acids. Break it down for us.
Okay, think of it like different metabolic pathways. Ketogenic amino acids, they can be broken down into ketone bodies, an alternative fuel source when glucose is scarce, like during fasting or a low-carb diet.
So if you're on keto, these are your amino acid buddies.
Precisely. Then you've got glucogenic amino acids. They can be converted into glucose, and some can do both. The mixed group.
So versatile.
Mm-hmm.
Are there easy ways to remember which ones fall into each category?
There are mnemonics like lamb and liver for ketogenic, that's leucine, lysine, and then honey, mango, vanilla for glucogenic, that's histidine, methionine, valine. But what's really important is understanding the why. For example, in conditions like diabetes, the body struggles to regulate glucose. Knowing which amino acids contribute to glucose production, well, it can be crucial for managing the disease.
That's a great point. It's not just memorization. It's about connecting these concepts to real-world health.
Exactly. Now let's shift gears to amino acid derivatives. It's incredible how these building blocks can be transformed into so many other molecules, other essential molecules.
Like a biochemical magic show. From hormones to neurotransmitters, even components of our blood. Where do we even begin?
Okay, let's start with phenylalanine and tyrosine. They're the precursors to the catecholamines dopamine, norepinephrine, and epinephrine.
Wait, those ring a bell. Aren't those involved in our fight or flight response?
You got it. They regulate, heart rate, blood pressure, even our mood.
So if someone doesn't get enough phenylalanine or tyrosine in their diet,
Yeah.
could it actually impact their stress response?
It's a possibility. These pathways are all interconnected,
Right.
and any disruption can have, cascading effects.
Makes sense. Okay, what about tryptophan? Isn't that the one we blame for that post-Thanksgiving food coma?
It gets unfairly blamed. It is a precursor to serotonin, which, plays a role in sleep and mood. But tryptophan is also the starting point for niacin synthesis.
And niacin is.
It's a B vitamin, vital for energy metabolism, a precursor for NAD plus and NADP plus SEC, which are essential coenzymes involved in countless cellular reactions.
Wow, so tryptophan is a multitasker. It impacts like both energy production and mood regulation.
Now, you mentioned a disease linked to tryptophan malabsorption, right? Hartnup disease?
Yeah. What is it?
It's a genetic disorder, impairs tryptophan absorption, leading to a niacin deficiency. Now, what's interesting here is this deficiency can cause pellagra.
Pellagra. I vaguely remember that from my studies.
It's characterized by the 3Ds, diarrhea, dementia, and dermatitis.
Whoa, so you're experiencing like severe skin lesions, cognitive decline, and digestive problems.
Yeah.
All because of a problem with absorbing one amino acid.
It's pretty alarming. It really highlights how a seemingly small biochemical imbalance can snowball into serious health consequences.
Absolutely, and it underscores the importance of understanding these intricate pathways and how they can be disrupted.
Exactly.
Okay, what other amino acid transformations should we know about?
Well, histidine, it can transform into histamine, involved in allergic reactions. Glycine is a precursor for porphyrin, a component of heme in red blood cells.
Interesting. So it's not just iron we need to worry about for healthy red blood cells.
Exactly. Then there's glutamate, a major excitatory neurotransmitter in the brain, which can be converted into GABA, which has inhibitory effects. It's also a precursor to glutathione, a powerful antioxidant.
Whoa, glutamate seems to wear many hats in the brain.
Yeah.
What about arginine?
Arginine is pretty fascinating too. It can be transformed into creatine, which plays a role in muscle energy, urea, a waste product our bodies eliminate, and nitric oxide, important for blood vessel dilation and various signaling pathways.
Amazing how one amino acid can have like such diverse downstream
Mhm.
effects.
It really speaks to the interconnectedness of our metabolic pathways. One small change can ripple through the entire system.
Speaking of interconnectedness, let's talk about branched chain amino acids, isoleucine, leucine, and valine. They're often touted for muscle building, but what makes them structurally unique?
Imagine their side chains having a more complex, like tree-like structure, unlike the simpler straight chains of other amino acids.
So they're the tree huggers of the amino acid world, preferring to stick together away from water.
Exactly.
Yeah.
And this structural quirk, it plays a big role in how proteins fold.
Okay, I'm starting to see how these seemingly small structural differences, they have like huge implications for the bigger picture. Now, you mentioned a condition linked to these branched chain amino acids. Maple syrup, urine disease. The name is definitely memorable.
It's caused by a deficiency in the enzyme responsible for breaking down those branched chain amino acids.
Oh, so it leads to a buildup of these amino acids in the body.
Yeah, and it causes much more serious problems too.
Like what?
Well, infants with this condition can experience intellectual disabilities and feeding difficulties. Early diagnosis and treatment are critical.
It really drives home the point that even a seemingly minor enzyme deficiency can have a profound impact on development.
Precisely. It highlights the crucial role of enzymes in maintaining metabolic balance. Welcome back to BiochemSerye. So far, we've categorized the amino acids. Now let's get to know each of the 20, individually.
Okay, I'm ready for a roll call. Where do we start?
Let's begin with the simplest one, glycine. Its side chain is just a single hydrogen atom, the smallest of the bunch.
So simple, yet so important, right?
Hmm.
Glycine's lack of a bulky side chain, it allows for like incredible flexibility in protein structures.
Exactly. It's often found in bends or tightly packed regions of proteins. Think about collagen, most abundant protein in our bodies, provides structure to our skin, bones, tendons. Glycine's flexibility, it's key for collagen's tightly wound triple helix structure.
So it's like the nimble gymnast of the amino acid world.
That's a great way to put it. Now let's move on to alanine. It's a step up in complexity from glycine, with a methyl group as its side chain.
And that methyl group makes it nonpolar, right? Hydrophobic, meaning it doesn't really like to interact with water.
Exactly. Alanine and other hydrophobic amino acids, they tend to cluster together in the interior of proteins, away from the watery environment of the cell.
Okay, that makes sense. Like oil separating from water, they want to stick with their own kind. Now, what about those branched chain amino acids we talked about earlier? Valine, leucine, and isoleucine. What makes their structure so special?
Well, like their name suggests, they have branched side chains, giving them a more complex, like tree-like structure compared to the simpler straight chains of alanine or glycine. This branching, it makes them bulkier and even more hydrophobic than alanine.
Interesting. So these bulky branched chain amino acids, they would really influence the three-dimensional structure of a protein, right?
Absolutely. They contribute to the protein folding process by creating those hydrophobic interactions we talked about, helping the protein fold into its correct shape.
Okay, starting to see how those seemingly small structural differences have a big impact. What about methionine? What makes it unique?
Well, methionine is the standout because it contains a sulfur atom in its side chain. Sulfur is highly reactive, so methionine participates in some important chemical reactions.
Okay, give us an example.
Well, for one, It plays a crucial role in protein synthesis. It's always the first amino acid incorporated into a new protein chain, like setting the stage for the rest to follow. It's also involved in methylation reactions, where it donates a methyl group to other molecules. These reactions are essential for regulating gene expression and a host of other cellular processes.
Wow, methionine's a busy bee. Okay, now onto the aromatic amino acids. First up, phenylalanine. Aside from being a precursor to those mood-regulating catecholamines, what does it look like structurally?
Well, phenylalanine gets its name from the benzene ring, also known as a phenyl group, in its side chain. Aromatic molecules like this have a distinctive ring structure. It gives them special properties.
Back to organic chemistry. I remember those benzene rings being very stable.
Exactly, and that stability plays an important role in phenylalane's function. It contributes to the structural integrity of proteins.
Okay, moving on to tyrosine. It's also aromatic, right?
Yes, it is. Tyrosine also has a benzene ring, but with an added hydroxyl group attached, making it a bit more polar and reactive than phenylalanine. It can participate in hydrogen bonding and other chemical reactions that phenylalanine can't.
So it's like phenylalane's more social cousin, interacting with a wider range of molecules.
A good analogy. And like phenylalanine, it's also a precursor to those important catecholamines we discussed earlier.
All right, now for tryptophan, our other multitasking amino acid,
Yes.
involved in both energy production and mood regulation. I remember it's aromatic too, but its ring structure looks a bit different.
That's right. Tryptophan boasts a double ring system called an indole ring, which includes a nitrogen atom. This unique structure, it influences its role in protein structure and its ability to be converted into serotonin and niacin.
So much complexity packed into that indole ring.
Yeah.
Okay, next on our amino acid list, proline. I remember this one being a bit of a rebel when it comes to protein structure.
Proline is unique because its side chain actually forms a ring with its amino group, which is part of the protein backbone. This creates a rigid structure that introduces a kink or a bend in the protein chain.
So it like disrupts those nice orderly alpha helices and beta sheets that proteins like to form.
Exactly. Proline's unique structure plays a crucial role in protein folding, but it also influences the protein's flexibility. Some proteins need those kinks and bends for their function.
Okay, that makes sense. Now onto the polar uncharged amino acids. Let's start with serine. What makes it polar?
Serine has a hydroxyl group in its side chain, which makes it polar and capable of forming hydrogen bonds with water and other polar molecules.
So it's like a tiny magnet, attracting those polar molecules. Where do we usually find serine in proteins?
Well, serine often hangs out in the active sites of enzymes. Those are the regions of the enzyme where the catalytic activity happens. In these active sites, serine can interact with the enzyme's target molecule called the substrate, or help facilitate the chemical reaction.
Okay, so it's a key player in enzyme function. What about threonine? Is it similar to serine?
Yes, threonine also has a hydroxyl group in its side chain, making it polar and capable of forming hydrogen bonds, but it has an extra methyl group, making it a bit bulkier than serine.
Got it. Now, what about asparagine and glutamine? What do they have in common?
Both asparagine and glutamine contain amide groups in their side chains. Amide groups are polar and can participate in hydrogen bonding, just like those hydroxyl groups we talked about.
So they're also hydrophilic, enjoying the company of water molecules. Where do they typically reside in proteins?
You'll often find them on the surface of proteins, interacting with the surrounding water molecules.
Okay, makes sense. Now on to cysteine. We've already talked about its disulfide bond-forming abilities, but
Yeah.
what else should we know about it?
Cysteine contains a sulfhydryl group, written as des-SH in its side chain. This sulfhydryl group is very reactive and participates in various chemical reactions. But the most famous one is definitely the formation of disulfide bonds with other cysteine molecules.
And those disulfide bonds are essential for protein folding and stability, acting like molecular staples holding the protein together.
Exactly. They can either link different parts of the same protein or connect two separate protein chains.
Okay, now for the charged amino acids. Let's start with the acidic ones: aspartic acid and glutamic acid. What makes them acidic?
Both aspartic acid and glutamic acid have carboxylic acid groups in their side chains, which are negatively charged at physiological pH. Remember, they readily lose a hydrogen ion or proton, giving them that negative charge.
Right, they're the proton donors we discussed earlier.
Yeah.
So how does this negative charge influence their role in proteins?
Well, this negative charge allows them to interact with, positively charged molecules or ions, influencing protein structure and function. These interactions are crucial for a wide range of cellular processes, from enzyme activity to signal transduction.
Got it. Now on to the basic amino acids: lysine, arginine, and histidine. What makes them basic?
These amino acids have side chains containing nitrogen atoms that can accept a proton, giving them a positive charge at physiological pH.
So they're the proton acceptors, the opposite of the acidic amino acids. What about their structures? What sets them apart?
Well, lysine has a primary amino group in its side chain. Arginine has a guanidinium group, which is a more complex nitrogen-containing group, and histidine has an imidazole ring, another nitrogen-containing ring structure.
So even within this category of basic amino acids, there's like quite a bit of structural diversity.
There is, and those different structures give them slightly different properties, influencing how they interact with other molecules.
It's fascinating how all these subtle structural nuances they add up to create such a diverse array of amino acids, each with its own personality and set of skills.
I agree. It's like they're all specialized tools working together to build and maintain the complex machinery of life.
Okay, so we've explored like all the individual personalities of these 20 amino acids, but I'm curious how they all come together to form those intricate protein structures.
It's like taking all those unique pieces of a puzzle and seeing how they fit together to create the bigger picture. The sequence of amino acids in a protein, it's like a blueprint. It guides the protein to fold into a specific three-dimensional shape.
So all those hydrophobic interactions, hydrogen bonds and ionic bonds we talked about, those are the forces that shape the protein.
Exactly. Imagine those hydrophobic amino acids like our branch chain friends, all huddling together in the protein's core, away from the water. Meanwhile, the hydrophilic ones, they're happy to be on the surface interacting with the surrounding aqueous environment. And those charged amino acids, they can form ionic bonds or salt bridges, further stabilizing the structure.
Okay, I'm starting to see how it all comes together, but there are a lot of weak interactions involved, right?
Yeah.
Are those enough to like actually hold a protein together?
They may be weak individually, but collectively they create a powerful force. It's like a team of ants.
Yeah.
One ant can't lift much, but together they can carry objects many times their size. There are also van der Waals forces, weak attractions between atoms that are very close together. These all contribute to the protein's stability.
So it's a delicate balance of forces,
Right.
creating this intricate protein structure. But how does that structure actually dictate a protein's function?
The three-dimensional shape of a protein is crucial because it creates specific binding sites, little pockets or grooves, where other molecules can fit like perfectly.
Like a lock and key.
Exactly. Those binding sites are lined with specific amino acid side chains that interact with a particular target molecule called a ligand. This interaction, it can trigger a whole cascade of events, leading to a specific cellular function.
So, for example, an enzyme might have a binding site for its substrate, the molecule it acts on, and the arrangement of amino acids in that binding site would be crucial for the enzyme's catalytic activity.
Precisely. Those amino acids are positioned perfectly to interact with the substrate and facilitate the chemical reaction.
We also talked about how amino acids can be modified, after they're incorporated into a protein, like phosphorylation. How do those modifications impact protein function?
Those modifications act like switches, turning a protein on or off, or fine-tuning its activity. Imagine like adding a phosphate group to a serine residue. It can change the protein's shape, altering its binding site or its ability to interact with other molecules.
So these modifications, they add another layer of complexity to protein regulation.
Absolutely. They allow cells to respond dynamically to changes in their environment and fine-tune their activities.
All right, before we wrap up, let's test your knowledge with a little quiz. 10 questions to see what you've learned about amino acids. Ready?
Bring it on.
Question 1. Which of the following is an essential amino acid? A, glycine. B, tyrosine. C, alanine.
The correct answer is B, tyrosine. Remember, our bodies can't synthesize essential amino acids, so we have to obtain them from our diet.
Question 2. Which amino acid is known for its flexibility and is often found in the bends of proteins? A, proline.
Hmm.
B, glycine. C, cysteine.
The answer is B, glycine. Its small size and lack of a bulky side chain, it allows it to fit into tight spaces and provide flexibility to protein structures.
Question 3. Which of the following amino acids is classified as ketogenic, meaning it can be broken down into ketone bodies? A, leucine. B, valine. C, histidine.
The correct answer is A, leucine. Remember, ketogenic amino acids are alternative fuel sources when glucose is scarce.
Question 4. Which amino acid contains a sulfur atom in its side chain and is involved in methylation reactions? A, methionine. B, cysteine. C, tryptophan.
The answer is A, methionine. That sulfur atom makes it a key player in various metabolic processes.
Question 5. Which amino acid is a precursor to the neurotransmitter serotonin, the tyrosine?
Yeah.
B, phenylalanine. C, tryptophan.
The correct answer is C, tryptophan. It's also involved in the synthesis of niacin, a B vitamin.
Question 6. Which of the following amino acids has a negatively charged side chain at physiological pH? A, lysine. B, glutamic acid. C, histidine.
The correct answer is B, glutamic acid. It's one of the two acidic amino acids.
Question 7. Which amino acid is known for its unique ring structure that introduces kinks or bends in protein chains? A, proline. B, glycine. C, phenylalanine.
The answer is A, proline. Its rigid structure disrupts the regular patterns of alpha helices and beta sheets.
Question 8. Which amino acid is often found in the active sites of enzymes and can participate in hydrogen bonding? A, serine. B, alanine. C, valine.
The correct answer is A, serine. Its hydroxyl group makes it polar and reactive.
Question 9. Which amino acid can form disulfide bonds with another molecule of the same amino acid, contributing to protein stability? A, methionine.
Yes.
B, cysteine. C, tyrosine.
The answer is B, cysteine. Those disulfide bonds act like molecular staples holding proteins together.
And finally, question 10. Which of the following amino acids is classified as glucogenic, meaning it can be converted into glucose? A, leucine. B, lysine. C, valine.
The correct answer is C, valine. Glucogenic amino acids can contribute to blood sugar levels.
That was a great recap. I hope you aced the quiz, and more importantly, I hope you gained a deeper appreciation for these amazing molecules.
I agree. Understanding amino acids is fundamental to understand biochemistry and human health.
It's been a fascinating journey into the world of these tiny building blocks. Thanks for joining us on this deep dive into amino acids.
Until next time, keep exploring the amazing world of biochemistry.